Okay, this podcast is going to be reviewing Unit 2, Part 1, which is all about cell structures. That covers standards 2.1, 2.2, 2.3, and 2.8. So in 2.1, we actually have two parts to this. The first part is to relate cell parts and organelles to their different functions. So the cell is the basic unit of structure and function for all organisms. It's one of the fundamental characteristics of life, is that all living things are made of cells. And most living things that we have talked about are eukaryotic cells, and that means that they are cells that contain a nucleus, okay, and have things called membrane-bound organelles. Okay. An organelle is the functional unit of the cell, so it's kind of like little organs for the cell. They do all the little jobs for the cell, just like our organs in our body do all our jobs for us. Membrane bound means that they are surrounded by one or two phospholipid bilayers, and those are membranes, and those are part of the lipid group that we talked about in the first podcast. So, in a eukaryotic cell, we will categorize all the different organelles by function. It's one of the easier ways to remember them. So, boundaries. The organelles that are involved with providing boundaries to the cell, you have the cell membrane and the cell wall. Remember, the cell wall is only found in plants and bacteria and fungi, some protists. The cell membrane is around all cells. Controls, these are the control mechanisms of the cell. The nucleus is like the brain of the cell, and inside the nucleus you have your various chromosomes, which have all of your DNA. That's all your genes, decides what proteins you make and everything. Assembly of organic molecules. We need to be able to make all of our different organic molecules, especially proteins, but also lipids, nucleic acids, all sorts of stuff. So involved in that, you have your ribosomes. Your ribosomes are going to be making proteins. Okay, your ER is also involved in that. ER stands for endoplasmic reticulum. There's also smooth ER, which is involved with making lipids, and everything floats around in the cytoplasm, and a lot of different chemical reactions will happen in the cytoplasm. Transport. You need to be able to move things around the cell and in and out of the cell. So the endoplasmic reticulum is involved with this. The Golgi apparatus will do this. And vesicles act like little taxi cabs. When things are made, they go inside the vesicles and they'll get transported all around the cell. Storage. Sometimes we need to store stuff. So vacuoles and lysosomes are good for storage. Vacuoles in plants are very large and store a lot of water. In animal cells, they tend to be smaller, but they'll still store water and food and things like that. Lysosomes actually contain enzymes, and so their job is once they're storing stuff, they can actually break some of that stuff down for the cell and get rid of it, and that includes worn out cell parts. You have organelles that are energy transformers, your chloroplasts and your mitochondria. Chloroplasts are going to do photosynthesis, mitochondria are going to do cellular respiration. We'll talk about more in the next podcast on those. Support, the cytoskeleton is a crisscross network of proteins all throughout the cell. That's especially important when a cell does not have a cell wall. So it gives the cell its shape, also allows for movement. Locomotion, also known as movement. So sometimes cells need to be able to move around. So they have projections that come off of them. The small ones are called cilia. You can have big long tails called flagella. And then cytoskeleton also can help with moving cells around. And then cells need to be able to divide. So animal cells use centrioles, and plant cells use centrosomes. And these are going to be special parts of the cell that are going to organize the spindle, which is involved in allowing the cell to divide. Now, one of the things MCAS likes for you to do is to be able to compare plant and animal cells. So plant cells, remember the big difference is they have chloroplasts, they have cell walls, and they have a large central vacuole. In animal cells, no chloroplasts or cell walls. Their vacuoles are small, okay, and they have things called centrioles that they use for cell division, like we just said, instead of the centrosomes like plants. Okay, so a lot of times they'll ask you to compare plant and animal cell. If you can remember these three things, you will be good. Okay, and here's just a picture of that, highlighting the different things between plant and animal cells. There's your cell wall. There's your centriole. Right, there's your chloroplast. There's something different. And then here's your large vacuole here. And over here, the vacuoles are very small in an animal cell. And this is showing you some of the other parts. There's that flagella that is used for movement. Plant cells don't have that. They have the cell wall. They all have a nucleus, endoplasmic reticulum. There's the central mitochondria in both, chloroplast only in the plant cell. So you can see the comparison between these cells. Okay, 2.1b is actually all about cell membranes and how they are selectively permeable. So when we think about cell membranes, remember all cells have plasma membrane, and plasma membrane is the other name for a cell membrane. In eukaryotes, you're going to find the plasma membrane around the outside of the cell, but also it's going to be around the nucleus. It makes up vacuoles, chloroplasts, mitochondria. This is where you're going to find all of the different cell membranes surrounding organelles. Also, cell membranes are actually what the endoplasmic reticulum, which is the ER, and the Golgi apparatus are made of. Functions of the plasma membrane. Controls what enters and exits the cell so that the cell can keep in homeostasis. So this plasma membrane kind of acts like a bouncer and lets certain things in and out of the cell and keeps other things from entering or leaving. Also, the cell membrane provides protection and support for the cell. So it gives it its shape, its structure, and gives it protection. So the structure of the cell membrane, you have your phospholipid bilayer. So you have two layers of these special molecules called phospholipids, and they look like they have a head with two tails. The inside here is afraid of water, so they orient each other because outside here is a water type environment, and inside the cell, the cytoplasm, is also a water environment. Okay, so it's protected on the outside, and then going through there you have various proteins. So this is a transport protein, so things can get in and out of the cell. These are carbohydrate chains that are going to be used for recognition. There's enzymes that are on the cell membrane, all sorts of stuff within the cell membrane. But these little phospholipids stick together so tightly that only things that are very small and that have no charge can get through them. So larger molecules have to go through these transport proteins. So passive transport is when we're going to move stuff in and out of the cell, and the cell is going to use no energy to make this happen. So molecules are going to move with a concentration gradient. That means they're going to go from where there's a lot to where there's very few, high to low. Okay, so molecules are going to move from high to low concentration. There's three types of passive transport. Diffusion is when molecules move from a high to low concentration. This could be any molecule. So if somebody sprays perfume on the side of the room and you can smell it on the other side of the room, that's because of diffusion. The molecules go from where there was a lot to where there's less. Facilitative diffusion is diffusion with help of transport proteins. Remember I said some things cannot get directly through the membrane. They're too big or they have a charge, so they have to have help, so they get help with proteins. And then a special type of diffusion is osmosis, and that is specifically the diffusion of water. We also have active transport. Sometimes the cell has to use energy in the form of ATP in order to move things across the cell. So it's going to actively move molecules where they are needed. And that is going to go against the concentration gradient. So we're going to go from low concentration to high concentration. So it's against what the molecules want to do, but sometimes we need them to do that. There's three types of active transport. There's protein pumps, where you're actually going to exchange molecules for each other, like the sodium-potassium pump. Endocytosis is where the cell actually looks like it's eating. So you get like a kind of indent on the cell, and it will actually take something inside of it. So that thing is on the inside. And then exocytosis is actually the reverse of that, where it would spit that thing out. Okay. Now also, when we put our cells need to be in homeostasis, and that has to do with osmosis and the amount of water that they're in. Okay, so if you put a cell into a solution where there is less water outside of the cell than inside, the water is going to want to move out of the cell, and that'll actually make the cell shrink. And if there's too much water outside the cell than there is inside, more water outside the cell than inside, the water is going to want to move in, and that can actually make the cell burst. So scenario A is when we have something in a hypertonic solution. That means there's more stuff dissolved in the water than there is water compared to the inside of the cell. We want our cells to be isotonic. Iso means stable, equal. And we don't want to be hypotonic either, where too much water goes into our cells and it can actually make them burst open.
Okay, now we are going to take a look at standard 2.2, which is going to take a look at the difference between prokaryotes and eukaryotes. So prokaryotic cells are your very small and simple cells, and they have very few parts to them. They have a plasma membrane, which is the same as a eukaryotic cell. They have cytoplasm, which is the same as a eukaryotic cell. They both have DNA. Okay, and they both have ribosomes. The big difference between a prokaryotic cell and a eukaryotic cell is that a eukaryotic cell has a nucleus, okay, and also eukaryotic cells have all of these things here, which are your various organelles. Okay, so prokaryotes, the only cell part that they have is the DNA and the ribosomes. So they can make proteins, but that's about all they need to do. Eukaryotic cells are much larger, much more complex than a prokaryotic cell because they have a lot more going on within them. Standard 2.3, we're actually going to categorize living things into the various domains and kingdoms. So classification, all living things get classified and named based on certain biological characteristics. So we're going to look at certain things about living things when we classify them. So we're going to look at their type of cell. So are they a prokaryote or are they a eukaryote? Okay, so pro or eu. We are going to look at some specific cell structures like cell walls, chloroplasts, things like that. Cell number, are they unicellular or multicellular? We would look at things like mode of nutrition, are they autotrophs or heterotrophs? And method of reproduction, do they tend to reproduce asexually, which is when they just divide in half, or sexually, where there is two parents involved and you get a mixing of your cells or your DNA. So when we classify things, we can classify them, first of all, into one of the three domains. This is the largest and most inclusive category of classification. And what we do when we put things in domains is we actually look at similarities and differences in DNA. And they ended up making two different domains of bacteria because they have very different DNA sequences. So the three domains are archaea, bacteria, and those are both domains of bacteria, and then there is eukarya. These two domains are made up of organisms that are prokaryotes, okay, and the domain eukarya is all eukaryotic organisms. Now within the domains we have 6 kingdoms. This is the next largest category of classification. The 6 kingdoms are Archaeobacteria, and that's with the domain Archaea. Eubacteria is with the domain Bacteria. And then there's 4 kingdoms that are part of the domain Eukarya. There's Protista, Fungi, Plantae, and Animalia. And this shows you some of the characteristics they have. Like Animalia are multicellular eukaryotic as well as plants and fungus, but there are other differences between them. And we can see that when we look at big classification. The other thing that we are going to take a look at are viruses. Viruses, this is standard 2.8, are basically simply a particle of nucleic acid surrounded by a protein. And the thing about viruses is they have to use a host cell to replicate. So they are not considered living things because they do not contain cells. They cannot reproduce on their own. They cannot get energy on their own. The host cell is the cell that gets infected by the virus and eventually destroyed by it. So the nucleic acid that is inside of a virus is either DNA or RNA, and then the inside of it, or that's the inside and the outside is a protein. So you have a protein outside, if I can say that correctly, protein outside, and then you'd have your DNA or RNA on the inside. And again, the virus has to infect a host cell in order to make copies of its DNA or RNA, and that's how it replicates itself. Okay, so the capsid is that protein going around the outside. So we can compare cells and viruses. So viruses have DNA or RNA, okay, inside a capsid. So the genetic code is similar, okay, but they can reproduce only with a host cell, whereas cells themselves can do independent reproduction. They cannot grow and develop. They cannot obtain and use energy like cells can. They do have the ability to change over time. Because they have DNA and RNA, they are able to undergo mutation, and mutation can lead them to evolution. That's why it can be really hard to fight some viruses.
And responding to the environment, they are unable to do. They either can tolerate the environment they're in or they can't, whereas cells are able to withstand and changes to their environment. So that's the end of Unit 2, Part 1.